position. I think it's recording now, yes. So
yes.
anyway, thank you everyone for being here. Thank you, Somenath, for accepting to be part of this seminar series online. I will share my screen to present the speaker. And again, I have to start from here. And I hope you can see the full screen probably. So today we have Somenath Bakshi, I hope the pronunciation is correct, from University of Cambridge. The title of the talk is Connective Dynamics of Antimicrobial Response Across Scales. And a couple of words regarding the speaker of today. So he's actually interested in systems and synthetic microbiology that inspect with different methods, molecular biology, single-cell microfluidics, time-lapse microscopy, and machine learning also. And the goal is to understand the emergence of antimicrobial resistance and tackle resistant infection via genetic engineering, so very broad, but he will tell you more during the talk. Some part of the CV of Somenath. So he did a PhD at University of Wisconsin-Madison, and after postdoctoral research at Harvard, and finally assistant and now associate professor at University of Cambridge. So with this one, I would like to stop sharing my screen, and so Somenath, thank you for being here. The stage is yours, and you can share your screen now.
Right. Thanks, Marco, and thanks, Rosalind, for the invitation. Thank you. I'll share my screen.
Yeah, we can see.
All right, perfect. So thanks again, everyone, and thanks for being here. And my name is Somenath, and we have recently established the Laboratory of Systems and Synthetic Microbiology at the Department of Engineering in University of Cambridge. So that's where most of my lab works happens, but some of the work that will present also is happening in a new shared facility I'm building together with a colleague, Mauri, which is called the Smart Microscopy Laboratory. And the idea of this facility is to run student-driven projects where we come up with sort of custom microscopy solution for a particular biological problem, which could sort of involve acquisition using 3D printed material and robotics, or could also be like a machine learning approach to extract more meaningful data from the images. And the website for that is not available yet, but I will present some of this work today. So the major focus of our lab really is to understand and control antimicrobial resistance in bacteria. I don't have to really motivate this, but just place the figure to show that it's a serious problem. There are more than 700,000 deaths directly linked to AMR during 2019, and the projection is that if nothing is changed the way it's spreading, then by 2050, sort of more than 10 million people will die, making AMR like a more serious killer than cancer. So a lot of the data are published in this AMR review conducted in UK. Unfortunately, the problem of antimicrobial resistance is not really preventable. Bacteria will always find ways to become resistant to the drug treatment. However, it is controllable, and that sort of is the direction that we are working towards. And the sort of 6 areas that are not all independent areas, but the sort of there are interconnection between them are these areas where my lab works. So we try to understand AMR from the point of view of emergence, where we focus on analysis of antibiotic persistence and heteroresistance. We try to understand how AMR infections often spread through plasmid transfer and how they get lost due to the plasmid loss, and also the evolutionary aspects of AMR from the perspective of cellular heterogeneity and fitness, cost, and benefit analysis, so on. And we often use this knowledge to develop strategies to control AMR, which is at the level of prevention, where we're engineering prebiotic phages and probiotic bacteria, urinary extracellular vesicles to tackle or to prevent infections. We also work at the level of detection, where we're engineering bacteria which can act as living diagnostics in our body, and elimination, primarily through the use of phage therapy and alternative antimicrobials. And in my talk today, I'll mainly focus on these 2 topics, antibiotic persistence and phage therapy. Now, these 2 sort of topics are kind of classified into these sort of systems microbiology aspect of our research, which is to understand the mechanisms underlying these problems, and using that information often to design synthetic approaches to tackle them. And these two sort of approaches are also often work hand in hand. We understand to engineer systems and often we engineer systems to understand them. So that's the core sort of theme of our lab. Now, the approach of our lab is really grounded at the level of cellular physiology. So we really focus on the cellular context. The reason being, we believe that often how a drug molecule interacts with the target is well understood, and how sort of the population level response happens for bacteria towards this drug can be well characterized. But to really understand the response of the population at short and long time scale at a mechanistic level, one needs to understand the cellular context, because it is the cell that determines that whether the drug can actually get inside, and whether it can reach the target, does it get pumped out, and also for the targets, like how many targets are actually expressed, how many of them are active, and what is the fitness cost of mutating that target. And so to really develop a mechanistic understanding of the response at the population level and the evolution of resistance, one needs to sort of understand these processes with the cellular context. And so we focus therefore a lot on understanding the cellular physiology and its impact on these processes. Now, due to various factors like such as stochastic gene expression and sort of really deterministic processes, but that can give to stochastic outcomes such as cell division and growth dynamics, we actually get a lot of heterogeneity at the population level of bacteria. So a lot of heterogeneity within a population at single cell level. And that heterogeneity of physiology changes with the environment. And so the response, if we want to understand it from the physiological perspective, we need to have a tight control on the environment and have single cell resolution to see physiological dynamics. So this is sort of where the methodological aspect of our work comes in, is that we try to understand physiological dynamics of individual cells in a tightly controlled growth environment. And we try to then connect those physiological dynamics from single cell level to the population dynamics of a short time scale and to the long time scales, which is sort of resistant evolution. So my lab have been, we have been developing these technologies to sort of address this goal. So we have recently developed a platform called Microcosm, which is where we can use a microfluidic device as an inline sensor and look at the dynamics of cells in a connected culture, but at a single cell level. And we have developed an inline sensor where the culture is continuously pumped through, and we have several optical sensors to detect the density and fluorescence of those cultures as the culture passes through. So we get continuous measurements without having to really stop the culture shaking, etc. These enables us to kind of link the dynamics of single cell level at the single cell level with the population level. But this is short-term dynamics. We can use the same sort of bioreactor, which we can use these inline density sensors to act as to implement a feedback control to maintain the culture at a particular density. And then we can use it to run evolution experiment. And we have engineered a fossil record collection system where a 3D positioning system continuously places samples in 96 well plate that are maintained at 4 degrees using a culture cooler. And that way we get a very detailed fossil record of these evolving systems, which we can analyze from genetic sequencing perspective, or we can use a barcoding technique to place multiple mutants in the same microfluidic device and quantify their changes in physiological phenotypes. And so that sort of is the integrated approach that we have developed in the lab. And I'll sort of present the motivation of why this sort of a new method was needed, why the existing methods are not suitable for this. Now, when we're trying to understand physiological dynamics of individual cells, throughput is an important thing, and which is why many sort of labs still use flow cytometry, because it provides a massive number of cells per second. However, the data from PACS does not provide an image of the cell, and the estimates of size from the backscattering measurements are not really reliable, and there are no information about the dynamics. So often snapshots are a good sort of a alternative to FACS, where people can do like high throughput snapshot imaging of bacteria. This provides image so of individual cells, and so one can quantify size, morphology, content of a certain protein using a reporter, etc. However, it still doesn't provide any information about dynamics. So one of the technology that has been used in the field for more than a decade now is tracking individual cells on agar pads, where a single cell slowly grows and divides and forms microcolonies, and these cells are tracked over time to quantify their growth dynamics, their gene expression noise, etc. But we found that there are a lot of artifacts associated with this method. But primarily, if you are interested in quantifying heterogeneity, the growth environment itself has to be uniform. However, when we look at sort of cell growth within this microcolony, we find that the food availability changes really rapidly at the center, while it changes does not change at the periphery. And depending on the morphology of the colony, you have a really non-uniform distribution of nutrient that is available to individual cells. So that means it's unless that is tightly controlled, it becomes very difficult to disentangle the factors that come from environmental heterogeneity and comes from intrinsic heterogeneity of the bacteria. Now, even if there was a good growth condition where that could be maintained, quantifying this heterogeneity becomes very challenging, and for optical reasons. So it's due to the fact that microscopes are diffraction limited, that if we try to image a cell with a diffraction limited imaging system, a significant part of the light actually comes out of the cell due to diffraction. So here is an example using a simulation where 6% of the light goes out. Now, if there are two cells next to each other, that means that some of the light will be allocated to the neighbor cell, while light from the neighbor cell will be allocated to the original one. And this creates a very serious problem when imaging microcolonies. So, for example, here is a simulation we have run where just using the same sort of amount of emitters per cell, we can show that individual cells are much dimmer, but when you place them in small and larger and larger clusters, they just appear brighter purely due to this bleed-through effect from light coming from their neighbors. So as the number of neighbors increase, cells just appear brighter, completely as an artifact of diffraction. And to test it, we ran experiments, and we found that that is really, that's also true in real experiment. You can see an individual is much dimmer than when it is a part of a big cluster. And these experiments were not done by letting the cells grow into a macrocolony. What we did is that we grew cultures and then plated them at a high density so that they instantaneously form clusters of various sizes. So these cultures, because they come from the same culture, they have the exact same distributions across the entire imaging field of view. But just by being part of larger colony, they appear brighter. And when we analyze the full data, we found that there is no overlap between isolated individuals and cells that are parts of larger colony, which really sort of affects the analysis. Not only at the level of this total intensity quantification, but it really affects noise quantification. So there's a nice illustration of this through simulation. You can see that if the true heterogeneity is very low, CP here stands for coefficient of variation, then convolving with the quantitative function results in an artifactual high CB, because really the intensity of a cell becomes a function of its position within the colony. And on the other hand, if the underlying CB is actually very high, then the diffraction effect acts as a smoothing filter and causes allocation of intensity from the bright cells to the dimmer ones and undermines the actual observed heterogeneity. And so you can actually observe the same CV in both cases, but they come from very different scenarios, and becomes very difficult to actually identify the sources of noise in such analysis. So to track to avoid a lot of these issues, what we use in our lab is a microfluidic device called Mother Machine. So which was originally designed by Suk-Chun Joon's lab in University of California, San Diego, in 2010. And in these devices, we have these really narrow trenches, and they're about 1.5 micron wide, where a single line of bacteria can grow and divide as they are fed from a media which is flowing orthogonally to the trench direction, which means there are no forces on the cell, and the cell can be imaged for a really long time. And because cells are isolated in their own trenches like this, they have no competition for food. So you can truly sort of study the intrinsic heterogeneity, and you can study them for a really long time to quantify the physiological dynamics of individual cells. And so we did a lot of optimization of this device in our recent paper, where we could eliminate any sort of food competition within the trench, and also ensure that the gaps between the trenches were optimized to ensure there is no diffraction bleed through, so that we can really reliably quantify the intensity of individual cells. And with this device, you can track cells for more than 100 generations, quantify various properties across the lineages, and within lineages. And from individual lineages, you can quantify the cell size and gene expression dynamics over time under spatiotemporal uniform conditions. So this really enables us to now study physiological dynamics and heterogeneity at a homogeneous condition. Now, we also often processes in sort of antimicrobial response especially involve a lot of rare events. And so throughput becomes limiting, which is why a lot of people use FACS. And so we wanted to sort of bridge the gap, and we made a version of this device where now we can track more than 132,000 individual lineages every 5 minutes. So this is the montage of the entire microfluidic device. And here's a tiny crop showing 230 individual lineages, and another tiny crop showing these are 4 lineages, each with 10 cells per lineage. And so you get a really large amount of sort of single cell tracking data, and enables sort of identifying rare events such as plasmid loss or persister cells or horizontal gene transfer. So we have more or less like a really good throughput, but also we can now study things at the level of dynamics. Now, one challenge we had to also address here was data analysis. Now, when we had this really high throughput, of course, we can't afford to kind of sit down and analyze the data ourselves manually. This images of bacteria, like the way we process this data is that we collect phase contrast images of individual cells, and we have fluorescence reporters for processes that we're interested in. And studying phase contrast and segmenting them for these small cells in these tiny devices is a really challenging problem. For one, you can see is that the contrast between the cell and the background is not something that one can use to really easily identify them from the background, because the device itself is darker than the cell. And what our goal is to kind of segment this cell outline to quantify intensities from these reporters. A big problem is also the fact that the phase contrast PSF is almost as large as the bacteria, which means if you ask a human annotator, they'll have a really hard time identifying exactly where the cell begins and ends. And so diffraction effects is makes it very challenging to segment, to even create annotated training data for training machine learning algorithm. So that was not an option for us either. And so what we did is that we built an end-to-end model for the entire process in our devices, where we simulated how cells grow and divide based on our knowledge of bacterial growth and division in these devices, and then simulated their 3D interactions with each other and the microfluidic platform. And from those, we extracted ground truth position of those cells and used a phase contrast image model to produce realistic phase contrast data. And so now we can train a machine learning model with realistic synthetic images, which are almost indistinguishable from real data. And the model learns the object-to-image relationship. And it can now see real data and produce very accurate masks. And it works really reliably. So on the right hand, this is a performance on a real experimental data. And you can see that it produces really accurate masks from this data. There are several advantages of using this sort of an approach of using synthetic training data to train machine learning models for image segmentation. First of all, it's really fast. It's about 1,000 times faster than human beings sitting down, and of course, like orders of magnitude less painful. It's also more accurate. So we saw that when, because we can now generate synthetic data, which comes with perfect ground truth, we can actually benchmark how sort of inaccurate people are when they're annotating data. So we asked several users, and when they annotated the data, we could see that they're always underestimating the cell size. Depending on the growth condition, their extent of error changes. And this comes from the fact that we're looking at a two-dimensional projection of a 3D cell, which causes it difficult to identify exactly where the contrast where the cell actually ends from looking at the contrast. And these inaccuracies in the ground truth data actually leads to inaccuracies in the performance of the machine learning model, because the model learns these inaccuracies. So when we took this model called Delta, which was trained with human annotated data, you can see the performance of this on a sort of a stationary phase cell here on the orange continuously fluctuates even though the cell is not doing any growth, whereas the result from SIMBAC is very precise, and this is plotted in blue, and gives a very precise estimate for actually cell size. And we collected our own data when we found that we can estimate the length of a bacteria with a precision of about 40 nanometer using these machine learning models. and the width roughly of with the precision of 20 nanometer. And so it provides a really, really accurate way of analyzing the data in an automated manner and in an unbiased manner. And it's quite a versatile platform. We can generate sort of images for bacteria in under various growth conditions for different species of bacteria, and also under sort of different imaging conditions and even fluorescence images. And it gives a really robust performance, so we can segment sort of any data from like a mother machine type of devices, two-dimensional colonies or agar plants to produce really accurate segmentation output. And so recently we have also used sort of SynBack as a virtual macroscopy platform to identify errors in imaging. And it's something we have submitted recently at a journal, but a version of it is available on online to see how our imaging and image analysis algorithms lead to different artifacts in quantification of microbiology from microscopy data. So this is sort of the end of the method part of the presentation. So I would like to ask if there is any question on the methods aspect.
So please, question from the audience, is there something?
Yes, hi. Thanks a lot for that part. Looks really interesting. I was wondering, so now you also always showed like a rod-shaped bacteria. Is this SynBack also able to produce different types of shapes, like for example, a simple cocci?
Yes, so I think the imaging platform can produce images of any kind of bacteria. There is no restriction there. Like, I mean, the image simulation platform. And so that should be able to easy to generalize to any sort of imaging platform or bacterial shape and so on. What is difficult for us to so far to generalize was the mother machine device itself. And so if it's a different platform you're using, then it's very easy for us to produce the data. But microfluidics wise, we are kind of now exploring a new design which uses suction in the device to kind of make really short trenches to grow cocci. But that's not there yet. But if you're asking about the image simulation, then yes, that is very much possible. Did that get
Thanks a
your
lot for the
question?
answer.
Okay.
We have a question from the chat. Maybe I can. Okay, two questions from the chat. I can read it aloud. So the first one is from Konstantinos. I have time-lapse experiments in family machines and having issues with phase contrast cell detection. Can I use a SynBack for family instead of mother machines?
Yes, I guess if you definitely. So thanks for the question. If you see the middle panel of this image, this is actually sort of bacterial images of bacteria in phase contrast when they're grown in family machines. And you can see that we could generate 2D images of cell cluster in similar environment. And that was enough to train the model to get really, really accurate output masks. So this should sort of be something that you can easily directly use for your application, I believe.
And from Ophelia, would SynBack work for lower magnification too? So times 40 or times 20?
Yes, so that is a very interesting question. We're actually, we did a performance benchmarking for sort of how the performance of SynBack depends on resolution. We find that down to 40x, it works actually really well. Below 40x, it becomes less and less accurate. And so what we're actually now developing is a super resolution network where we have the same ground truth and we generate images with two different resolution, and we train a super resolution network with that model. The nice thing here in mother machine is that we can actually exploit the symmetry of the device, where there are vertical boundaries between cell and the device and horizontal boundaries between two different cells. So the model can accurately learn these features from sort of seeing two different images of two different resolution. And so we can actually go to 20x data and upscale it using the super resolution network and then segment them. So yes, any sort of down to 20x, we can definitely work. We haven't tested it on 10x. We're interested in trying that. But 20x was sufficient for us. And if you're interested, yes, definitely send me an email for the person who asked about the family device. Me and my student, Giorgio Sardo, we can definitely sort of help you with this.
Thank you. We have actually another question from Vinka. If I mix culture of different species, can the SynBack work for accurate segmentation?
Yes, so it's versatile enough. So it won't be able to identify. We haven't sort of trained a discriminator model yet, but it is possible. Like if you know the morphology, it can even discriminate between two types of sort of bacteria from the shapes. But as you can see from this image in the middle, there are occasionally like filamentation in this bacteria, and it produces very accurate sort of mask for filamented cells or like cells that are really small. And so it doesn't, because we train the model with quite a diverse set of synthetic data and in a very large, with a very large volume of data, the model is very sort of generalizable. So if you had mixed cultures, it can really do it. So we actually, in the SynBac paper, we demonstrated how it can continuously track and segment cells from, as the cells grew from exponential to stationary phase, where they were like 14 times smaller in cell area. And so it is really adaptable. So I think if you have mixed culture, it should be able to segment them.
And a side question from Justice, I don't know, do you have any co-culture work? So something that demonstrates co-culture?
Not yet. We are making a sort of a side-by-side trench mother machine to grow two sort of types of strain next to each other. But at the moment, most of our co-culture work is where we actually grow the two cells in the flask and flow the conditioned media into the mother machine. We haven't done microfluidic co-culture yet.
Okay, I don't see any other question from the chat. If you don't want to write in the chat, actually, you can open your microphone and speak it out, but it's fine. If no one has other question, I think we can go on.
Okay.
Yeah,
Thanks again. All right, I'll now sort of go into some of the illustration of these approaches. So the first application I'll talk about is about spontaneous persisters. Spontaneous persisters are these very rare cells that appear in a population of bacteria, which happen to sort of enter in a state of dormancy. And therefore, when you treat them with antibiotic, these cells can just survive or persist through the treatment. And when the antibiotic is removed, they can go back to recreate the original population because they are not genetically any different. They're simply a phenotypic outlier. And it has been very difficult to study these cells because of this transient nature, because you have to kind of catch them during the act, because they're going to just go back to recreating the original population. And they're very rare, so they happen, the population estimates were that they are actually appearing in less than 10 to the minus 4 per generations. And the other problem in trying to study them using a culture or like a population technique is that they are very slow growing, which means all the other sort of fast growing population will very quickly outnumber them. So you can't sort of track them over time, and so they get very, very quickly outnumbered. Now, this is where the microfluidic device mother machine provides a really massive advantage. First of all, we have the throughput to be able to detect such events because we can look at 10 to the power 7 generations per day. So such rare events, and we can still get like hundreds of events per day to get enough data for good statistics. Mother machine also eliminates competition, which means that if in one trench a persister appears, it is not affected by the fast growers in the other trenches. So we can have extended studies of how cells enter persistent states and then how they exit those states. And of course, because of the nature of these studies, we can actually follow the dynamics of entry, exit, and the physiological dynamics during this process. So here's going to be like this is a new study that we're doing, but here's a clear data to show how we can now detect persister, and we find that they're actually not completely dormant. They can actually continue to grow. They are 3 times slower in their growth, but they're very stressed. So they have 50 times higher amount of the stress response sigma factor, and they appear only 1.5 times 10 to the minus 5 per generation with this frequency. And so you can see that in this experiment, which is movies looping through, around 25-hour time point, we flow antibiotic, and all the other cells immediately start to die, whereas the entire population actually can survive this lineage. And we find that the residence time of this particular phenotype is about 14 generations, and then they switch back. But that resident time was dependent on the RPOS gene. So if we knocked out RPOS, then we saw that while the entry frequency did not change, the exit frequency changed a lot, and so their residence times were much shorter for only 3 hours. So this is an illustration where this kind of high throughput devices with automated analysis can actually enable us to study what are the physiological markers for sort of type 2 persister or spontaneous persisters, and now we can sort of look into the gene regulation mechanism that underlie the emergence of these types of cells.
Sorry to interrupt you. We have a
Yes.
question from Philip Glaser, if you'd like to speak it.
Yes.
Yeah, so thank you. This is very interesting. But how you show that these are true persisters and not like some heteroresistance, for example?
Yes, that's a good question. So at least what we are doing at the moment, so we're trying to sort of use an optical tweezer to grab these cells out and do genetic sequencing on them. So if we get enough, I think then because they're transient, so we have to get large amount of cells to do the genetic sequencing. But because they're going back to the sort of the like normal sort of physiology within 14 generation, our initial assumption is that these are going to be sort of spontaneous persister, but yes, it is likely that they could be heteroresistors. But I'm not really familiar with the time scales of heteroresistance is also transient, but I don't know the time scales of how long those states are usually stable for. Don't know if you could provide some insight in that.
Yeah, thank you. I think
Thank you.
Okay.
Yeah, thank you.
Okay, thanks. And the other sort of persisters that are also really curious and interesting to study are type 1 persisters, where sort of bacteria in nature and in your body, they go through sort of periods of sort of good food and no food. And often in the starved condition, bacteria enter into, a certain fraction of bacteria enter into type 1 persistence or triggered persistence. And so we wanted to study such processes. However, Mother machine was not suitable originally to be able to study that because the environment is also temporally uniform. And what we wanted to do is to kind of do bulk culture growth curve, but with the mother machine platform. And so basically we wanted to have a system where we could track individual cells as they're growing in a culture. And the idea we had was quite simple, which was to hook up a culture with the mother machine. And then as the cells from the mother machine come out, they inoculate the culture. And the entire process is hooked up in a way that in a way that the cells from the culture deplete the media, and this depleted media flow through the device, and the cells in the mother machine then responds to the depleted media as if these cells are actually growing in the culture. So you have sort of the mother machine as a connected array of traps where individual lineages are trapped. And so we could study the dynamics of these cells at single cell level to understand what's going on in the culture. Now, while the idea was simple, it was very difficult to implement it because coupling a shaking culture with a system where you're trying to do very high resolution microscopy required us to sort of really separate the vibrational component of the system. And also we wanted to make sure that the starvation is actually uniform in the device. The microfluidics had to be redesigned so that we have enough retention time. And biofilms and bubbles cause a lot of problem because we are shaking culture. And we also wanted to reduce delays from the entire process. We wanted to really study things in the culture, not in the tube, and so on. But we have kind of addressed it now. And so I think now I can present to you like a data where you'll see on the top panel sort of how individual cells respond and behave as they go through repeated cycles of entering stationary phase and exiting. So at the bottom panel, I'm plotting the optical density of the culture using an inline sensor. So you have 2 cycles here, and you can repeat it as many times as needed. And on the top panel, you'll see 3 lineages that we picked to show this, where individual cells have a stress response marker on them. And so if you start the movie, at the beginning, the optical density is very low, so cells are not stressed. They're dim, they're large, they're growing really, really fast. And as the optical density starts to climb up, the amount of food per cell drops, and you'll see the cells are actually getting slowly smaller and smaller, and eventually they start to stop dividing and get very stressed and very bright at the sort of late stationary phase. And then after 9 hours, we switch the media to fresh source again. They quickly wake up to restart their growth and exponential sort of nature of this exponential growth and division, low stress. And then as the food runs out, they go back to these states again and show bright stress response marker and stay stationary until sort of we flow fresh media and restart the exponential growth again. So this way we can really study the entry and exit dynamics at individual cell level with various types of genetic and physiological markers. Now, the kind of data you generate from these is here. So on top, we have just one lineage shown here, which is like where you can see the individual cell is growing and is large, low stress. And then during the high optical density, which is plotted in black, cells become tiny, really bright from the stress response marker. Things go down again and up again as the optical density. So they really track it. But it also shows nice heterogeneity. So for example, you can see this orange stress where the cell was really highly stressed in the first round of stationary phase. In the second round, it actually had quite a low stress response, whereas the red stress, where the cell was one of the lowest in the stress response level in the first round, actually had a really strong stress response in the last round, in the second round. So it shows that these are sort of purely phenotypic sort of response, and they create quite a large variability in the stationary phase at the level of stress in individuals. And in the blue traces, we're plotting the growth of individual cells, and as they sort of enter stationary phase, and then when they wake up. Two sort of interesting features that we learned from here, for example. One is that we see how actually cells become small in stationary phase. We found that cells actually do not slow down their division process due to a phenomena called rate maintenance. So they keep dividing at the preset division time. It's only that they're adding the biomass much slower, which causes them to become really quickly small. And same thing happens repeatedly every time we do this experiment, and it comes from the memory of the replication fork, which causes the division to kind of proceed with a set time point. Now, when sort of cells exit, we see that often some cells exit very quickly, and we can trace it back to the fact that those cells did not divide one division when they're entering stationary phase. On the other hand, cells that do divide, they actually become small and they exit much later. Right, and so there is this trade-off about how many divisions you had before and after. Now, before we get into the, sorry, is there a question?
Yes, Somenath. So
Yes.
I have a question regarding the previous slide. So
This one?
the expression, what you are showing here in the GFP, are these cells are coming from the plasmid, or it's a chromosomal integrated ones?
It's a chromosomal integrated report.
Okay.
Yes.
So, okay, sorry. Yes, thank you.
Yep. Now, before we sort of go into the interpretation of the result, we also first wanted to validate that are we really seeing what's happening in the culture? And so what we did is that we developed a second method to actually snapshot bacteria from the culture continuously and compare them with the dynamics that we're observing in the mother machine. So on the right hand, you can see there are the continuous lines are for data from mother machine, and the snapshots from this second experiment, which are done at the same time, are plotted as these dotted dots with error bars. And we can see that the data in the mother machine actually follow really nicely to things that are happening that we observe from the culture directly. And we had a microfluidic platform sort of developed called MAX, developed by my colleague Burak Okumas, where we could actually continuously snapshot them during the exit phase, and we could see that the data from the snapshot really closely tracked the observations from the continuous measurements. So it means now we can get continuous data from sort of about cell growth and division and stress response and other reporters when they are going to these cultures. Now, I mentioned about this trade-off that if a cell divides sort of early before it goes into interstitial phase, it wakes up later. And if it does not divide, it actually divides when it is exiting stationary phase. And we found that this sort of phenomena kind of cancels itself out when the stationary phase is kind of the average stationary phase, which is like an overnight of like 12 hours of stationary phase. And so on the brown data is plotted for the cells, the number of division during entry, and for the green ones are plotted for during exit, and you can see they kind of cancel together. So the total amount of division actually is independent of the strategy. So this sort of heterogeneity in the way the cell enters and exits actually didn't matter in very short stationary phase. However, we found that if we actually keep the cells in stationary phase for prolonged duration, the smallest 10% of the cells actually could not even wake up. So it's the only top 10% cells that had 80% success in waking up. And if you plotted the chance, the probability of dividing during exit after a sort of a 3-day long stationary phase, we found that the probability was monotonically dependent on the length of the cell, which meant if they're smaller, they wake up much sort of, they're less likely to wake up. And if they're larger, they're more likely to wake up. So here, somehow this heterogeneity kind of had a one direction where there was a benefit. If they're large, they could wake up really quickly. On the other hand, if we did antibiotic treatment, we actually found that it is only the small cells that can actually survive antibiotic treatment because they don't wake up. So here is one example trench where at the bottom you can see a very small cell that did not wake up during the antibiotic treatment. And when the antibiotic is removed, this cell sort of resumes growth and repairs the population. And we found that it is only these small cells, these orange ones that are in stationary phase, that did not wake up, which would actually persist the antibiotic treatment. The large ones woke up and just got killed. And so we found that when we tracked about 20,000 of these lineages during exit dynamics, only 200 cells were persisters, and they were all in the small class of cells. Interestingly, we found that less than 10% of the small cells would actually persist, which meant that just being small was not enough of an indicator that, oh, they're going to be slow and therefore won't be killed. And so we did a follow-up study with Tim Lewis's lab in Northwestern University, and we found that using an ATP sensor, that only among the small cells, only the low ATP ones are actually the ones that could persist the antibiotic treatment. So here in these data sets, when we studied the really small cells, only 16 out of these 800 cells were actually persister, and they all had really low ATP levels. And so now sort of exploring exactly what kind of processes underlie the emergence of these low ATP cells, and exactly sort of how we can kind of come up with identify phenotypic markers for the rapid detection for them. And we're also exploring ways of testing anti-persister strategies to eradicate persisters. So that's sort of the second part of my talk. And so if you have any question, I can answer them now, and then I'll get into the last section of my talk.
Any question from the audience, either in chat or from the microphone? We had already a couple of questions from the audience during the talk, so let's see if someone would like to have it now. Apparently not really. Maybe you can go on with the next one. Oh, wait, someone pop up.
All right.
Okay, what was the pH during the experiment is asking Justice. And
so yes, that's a good question. So actually during this experiment, we can set the starting pH to 7.2, but actually the pH changes during growth curve experiment. And so we found that when cells are entering the mid-log phase and eventually to stationary phase, initially the pH actually drops and becomes quite acidic. And after the diaphysic shift, so it becomes acidic down to levels of like 5.8. And then after the diaphysic shift, cells actually, the population starts to increase the pH and goes back near the normal level, 6.9 and so on. So it's difficult that because we don't really sort of maintain the pH externally. The experiment goes through these sort of two phases of decrease and increase in pH.
Thank you. Rosalind, please.
Yes.
I was wondering about the connection between being a small cell and being a persister. Or do you have any idea why being small should kind of help with that?
Yeah, I think one hypothesis we had was that that if you are small, then you're likely to have you're going to have smaller number of things. So here, events where the actual concentration wouldn't matter, but the copy number would matter. Then if you are small and you have small number of things, you're more likely to have zero number of things. And so I think so that would be a hypothesis. Yeah.
Okay, thanks.
Yeah.
And I think when Justice is again chatting about pH, don't you think that it might play a role on making cell persistence?
It definitely does. I think there is a really nice paper published in 2020 where we see that, well, not us, it's actually another team who saw sort of genes involved in acid regulation, acid response called GAD-A. They're all sort of also involved in the persister phenotype. So I think there could be multiple sort of genetic and metabolic factors that underlie the emergence of persister. And so which is sort of why we did not want to use mutants to sort of amplify their number and see so that we could detect them and study a process, because it might just undermine one of the underlying processes. So at least now that we can detect it, we can use different kind of reporters to study exactly how they affect the total population. And yes, acidic response is definitely one of the sort of players here.
Thank you. And Gielina is asking something regarding the cell size and ATP. So did you see large persister cells with low ATP?
We did not. So actually, if you see, this is only for triggered persister. So I cannot comment on the spontaneous persister, which we see appearing in the exponential phase. But in the triggered persister, more or less all the cells are really small. And among them, the ones that we saw that could persist are actually from the smaller 25% quantile of the cells. So we did not see large stationary phase cells to have low ATP.
No other questions?
Okay.
Okay, so thank you a
Thanks.
lot. You can go on. Thanks.
Thanks. So that in the last phase of my talk, I'll talk about a new sort of research direction that we're taking to understand how sort of phage infection is also affected by the physiological heterogeneities of bacteria. And like an antibiotic, like every single step of a phage infection process is really strongly dependent on the physiological nature of the bacteria, because it depends. Phages are obligate parasites, so they really rely on the host resources. So here is a schematic that shows like how like the injection process, maybe even the absorption due to the membrane properties, the production of new DNA, the production of the capsids and assembly, all of these steps are critically dependent on the cell physiology. So our question was that since we know host physiology is going to be very heterogeneous, how noisy are these individual steps of phage infection? And what is the role of this noise in population dynamics of phage bacteria systems? So we kind of started the process by first asking the second question to see how we can motivate this study. And so we started with sort of playing with what is the impact of noise in lysis time. So using computer simulation, we set up an assay where an experiment where we're simply taking cultures of bacteria, inoculating them with phages, and then as the culture gets eliminated and the phage population goes up, then we get diluted, and then we sort of regrow the culture and add phages again. And so these are simulated traces of bacterial population collapse and phage growth. And from this data, we can quantify two kind of sort of properties of the phage population. One is what is the rate of growth of individual phage population, and if we mix two phages together with different properties, then we can quantify what is the competitive advantage of one phage over the other. And so by quantifying their relative abundance at the end of each cycle. And so when we did that, we found that actually noise can really make a big impact. So for example, there are two populations of phages being compared where we have the mean lysis time of 20, whereas one has a standard deviation of 1 and the other one with 3. And we found that actually as we increase the standard deviation of the second population, both in the metrics of the growth based competitive advantage and also this SCOMP, the competitive advantage at the end in abundance, they always win over the more tightly sort of phage with a more tightly distributed lysis time. And even the shape actually matters. So if we fix actually the mean and the standard deviation, but change their shape, we find that the ones with the lower, more tail to the lower values actually win over as we increase the variance. So it kind of motivated us to sort of investigate like that. It seems that noise in these processes are going to really have an impact at population dynamics and possibly can be selected for in evolution. So how can we quantify that? And so we then sort of course, went back to mother machine, because here again, like before an infection happens, every cell is experiencing identical environment, which means we can really kind of quantify the infection kinetics across different events and put them together to kind of understand exactly how much heterogeneity there is. But also at during these infection events, we have the full physiological history of that cell, and so we can understand how the physiology of the bacteria affects the kinetics of these steps. Now, a regular mother machine was not useful in this study, so we had to design a new type of a device where next to each mother machine trench, we have a very shallow trench. These trenches are only 300 nanometer tall, and these are needed for the phages to diffuse through and infect the mother. So the study I'm going to show is going to be, this study at least was done with E. coli wild type and then wild type T7 and the T7 level with YP gene at the capsid promoter done by our collaborator Diana Fusco's lab in physics. And so the really nice advantage of these phages is that every time a cell gets infected by one of these phage, the cell eventually glows in yellow fluorescence. So you can actually detect them. And using this, we could benchmark different microfluidic devices, and we found that only devices which have really wide trenches are actually suitable. So if we compare the fraction of trenches that had any infection or the probability of getting infection to the mother and then eventually cleared out due to lysis, it is only the blue design that enables really high proportion of like near 100% infection rates of getting the mother infected and getting clearance. So that's why we opted for a new design of these 3-layer microfluidic devices. So here I think I'm going to show you probably, I don't know if you have seen, I don't think it exists before our study, to be able to follow individual infections in a time-lapse manner. So this will be a movie where cells are growing in these trenches continuously. And every time you can keep your eye on this trench. Whenever a cell gets infected, it will first go glow in yellow, and then it will pop, releasing viruses, which will go and infect all the other cells in the trench and eliminating them one at a time. So let's start the movie. Let's see the first cell got infected. Here's the trench. And so these are 30-second resolution movies where we can study sort of hundreds of such trenches over time. And one nice thing sort of, I'd like to maybe, if I replay this, oops. You can see that this trench actually got an infection right at the bottom. Yep. So that infected cell could not produce phage to infect to within the trench, and therefore that trench was never infected. So which means that every trench, even though they're producing of the order of thousands of phages, they are completely independent. There are no cross-contamination between trenches. So this assay can be really nicely used to kind of study heterogeneity, but also screen different types of knockouts towards the same bacteria. So that was a really fast movie, but I'll play it slowly for you to be able to follow the steps. So I think this is the cell that's going to be infected first here. And so it becomes yellow, really bright due to the expression of capsid marker. Lysis produces phages, which then go and infect all the other cells in the trench. And then sort of just stochastically, the cells just pop over time. And so from each of these trench, we can quantify how sort of the cells get infected, and then eventually at what time points they lyse away. And you can see that during the first infection, all the other cells actually continue their exponential growth and division. So you can really treat the second round of infection as a stationary population. So you can really compare the heterogeneity at the lysis time. And using SIMBAC and machine learning, we can then process this sort of data for every single infected cell, and we can quantify their growth and division before infection, then exactly when the growth halts due to the infection, and when the capsid gene expression starts, and when the cells lyse. From this data, we could quantify various sort of phenotypes or various markers of the infection process. So you could see exactly when the growth arrest happens, when the production starts, and when the cell lyses. We found that roughly about 10 to 11 minutes before lysis, the cells actually stop their growth. And sort of 2 minutes after that, they start production of the capsid. And so there is that delay between sort of taking over the resources from the host and then starting to produce its own gene expression. But again, we're focusing on the capsid, which is one of the late genes. But our results kind of starts that to show, for, at least for T7, that there might not be as much distinction in timing in what people call late and middle genes. Now,
sorry if I interrupt you. There is a
yeah,
question
please.
regarding the slide before, and some
Yes.
a technical question from Dutta. How do you
Mhm.
infect the fourth cell in each channel?
How do you infect?
The fourth cell in each channel.
Oh, I
So,
see. So the assay basically is that we flow phage lysates in the mother machine at the main trench, and we flow it a very low MOI that only one phage is likely to go up into individual trenches. So we just wait until one of those phage swims up and infects a mother. So it's a purely random process, and we just take the movie for 15 hours and then analyze it afterwards. So I hope that explains maybe.
I suppose so. Yeah, thanks.
Okay. Thanks. Where was I? Yeah, I was here. One thing we first found was actually that this host takeover process, where sort of the growth of the host continues exponential growth right at the time until when the growth, the phage takes over and just the growth, basically growth will become zero. is actually quite reliable across infection events. So different cells here, we have plotted 23 of them, and you can see that they follow almost similar pattern of how their growth was before infection and after infection. And within 2 minutes, they go from full speed growth, which is first consistent with 22-minute doubling time, to like zero growth. So that while that was consistent, the amount of capsid protein produced per cell was really variable. So there was an order of magnitude difference between cells. So there are some cells that produce very few and some that produce a lot. If you plot it in the log scale, you could see that this variability actually came from partly when the cell lysed, but we also found that a lot of it was due to the size of the host they infected. So the larger the cell was, the more amount of capsid it produced, purely because possibly it has more ribosomes and other resources to make proteins. And so the correlation of between the cell size and the final capsid amount protein produced was pretty high. And if we color the data now in the sort of size of the cell at the point of growth halt, you can see them, it kind of nicely ranked the outcome. So one example where physiology of the cell really impacts the outcome of the phage infection process. Now, so far we have kind of talked about sort of the process after host takeover and to the until lysis, but we haven't really talked about sort of the first steps. And so before that, I'll show one thing is that even when we went down to sort of 1 second time resolution, we couldn't see really the lysis process. Basically, the cells disappeared within a frame. There was an intact cell, and the next one there was nothing. And so we couldn't really see a detail of what's happening when the cell is lysing. But this data was really useful for us to see that actually when we look at individual lineages, we found that individual lineages have much lower variance in lysis time. But it was about 70 seconds, even though the variance was actually larger between trenches, which meant that individual lineages have more similarity in their physiology than between trenches. So to be able to see the final step, we actually ran movies at a very high time resolution. So these are data collected at 10 millisecond time resolution. And at that resolution, at this resolution, we can actually see the lysis process. So here you can see a cell that is infected by phage, and it's just the last few seconds we are looking at. And so right around now, you can see there is a black cloud coming from the right of the cell. And the cell is popping and losing its contrast, and the phage gets released to these environments. And so here is probably easier to see if you see this montage, the chymograph. You can see this is where the cell pops and releases the phages, almost as a black cloud. And when we quantified the contrast of those two images, we could see that this entire process takes only 300 milliseconds, at least for this virus, and which suggests why we couldn't see them even at a 1 second time resolution. But now we have a really nice way to sort of see exactly what is happening, like the mechanical processes involved. We could probably dig deeper into this step as well. Now, we wanted to then go back to time zero, like what happens when the actual process starts. So what we did is we labeled phages with a DNA binding dye. So if we soak the phages in a DNA binding dye, the dye gets into the phages and coats the DNA. And so every phage becomes bright orange. And so with that, we can see exactly when a phage lands on a bacteria, and we can also follow the genome induction process. So here in the movie, we're going to see a phage land on the second cell. There it goes, and it gets dimmer. And so that cell is doomed. So if we just keep following it, you'll see right around 18.6 minutes, it will disappear. So there you go. Right? And so if you track those processes for individual cells, we can see this is when the phage actually landed. And because we're taking a much higher time resolution data, it's a 3 second resolution, we can see how the phage signal goes dimmer over time. So for each of those traces, we can see this is the first phage landing and a sharp spike, and then the signal goes down over time. The second phage landed, which was much brighter, and we think it's a damaged phage because it doesn't inject, but shows also that the decaying signal is coming from injection, not photobleaching. That phage, and when the cell pops, the membrane is permeabilized, so all the dye in the environment actually comes into the cell, so the signal goes up. But from this kind of data, we can quantify actually the absorption time and also the time of genome injection. And we find that it's on an average 5.6 minutes for the phage to inject the DNA. Interestingly, we found that there are two classes of injection. There are phages that do very gradual injection, and phages where the injection starts gradually, but then has a very rapid sort of kinetics when it gets all the DNA injected at once. And we're now investigating the sort of possible sources of this heterogeneity. But with this, we actually now have all the time scales, so we can actually see when a phage lands, injects its DNA, then at what point the cell growth is halted when the capsid gene is expressed. So we can first time actually have not only the timing and the associated variation at each of these steps. So how long it takes for the genome to be injected, for the growth to be halted, capsid production to be started, and when the cell actually lyses. So we can start a way, sort of an approach to kind of screen engineered and natural phages with kinetic profiling of the processes to see exactly what changed and what's different. And it also will help us now to analyze phage bacteria coevolution in a very detailed manner. So with that, I'll stop and I'll thank the people involved in this work. So I have an excellent sort of group of students in my lab. Georgios did the work on synthetic image generation and machine learning. Charlie is the student who led the phage imaging project. Erez is one of the a very young student, he's very talented, he's doing all of this lineage tracking process because it's very tricky when cells continuously disappear in this phage infection process. So you have to develop new algorithms for that. Naparna, Jack, and Aaron are new students who are also going to work on these areas. Alma is a genetic engineer and student currently a postdoc in our lab. And we're supported by two excellent sort of technicians, Nargis and James. I'm also thankful for a great sort of group of collaborator, Jade Wang in USA, my previous postdoc advisor, Johan Paulsen, Diana Fusco, with whom we collaborated on the phage imaging project, and then Fernanda Pinheiro in Milan, Ashraf in Cambridge, and Tim Williams. These are also our new collaborators on these projects. I will take this time to also thank the people who funded us, and I'll play the movie and thank you for your attention and happy to take any further questions.
Thank you a lot for the very nice, impressive talk, Somenath.
Thank you.
So I'll